All right, so the next procedure we're going to record data for is the effect of temperature on enzyme activity. And as you can see, we are going to take a look at the table right here. We have test tubes number 7, 8, and 9. And the contents of all 3 test tubes are the exact same. They're all amylase and starch. And we know that amylase and starch prove to have a reaction because in our past 2 data tables, we had amylase and starch producing color results in the Benedict's test, which means that there are sugars produced when amylase is breaking down starch. So now knowing that, we're going to be seeing which temperature is amylase the best at breaking down starch, right? So we're going to look at 0 degrees, which I had an ice bath for. We're going to look at 40 degrees, which is around human body temperature, and we're going to look at 100 degrees, which is boiling point of water. Okay, and these are all Celsius. So I had 3 separate test tubes. I had test tube 7, 8, and 9. And they were all in separate environments. And as you can already take a sneak peek, they're all changed in color from the Benedict's test, but there are slight variations in their test. So I'll get into detail when I actually show you. So you're going to be recording the color of 7, 8, and 9. Remember that their temperatures are what is different now, right? The contents are exactly the same. So in test tube number 7, at 0 degrees Celsius, we have this orangey yellow color. So you can see there's a little bit of separation in there, and it's not really a dark orange, and at the bottom it's still kind of blue, right? So it's this lighter orange, almost orange yellow color. You're going to tell me, is there sugar present? Right? When you're mixing blue and you're mixing orangey brown together, it's going to come out somewhere in the middle like this. Okay. Test tube number 8 was at body temperature or 40 degrees Celsius, and I'll hold them up together actually at the end to show you, but this is a darker true orange color. So you're going to tell me, is there sugar present? And what is the conclusion? Did the amylase break down the starch or not? And number 9, test tube number 9 is at 100 degrees Celsius and boiling water. And here we see that it is a true orange as well. Right, so it's completely orange through and through. No blue left over. So tell me, is there sugar present? And tell me what that means in terms of the starch being broken down. So I don't know if you can actually tell, but the one over here for test tube number 7 is a lighter orange than test tubes 8 or 9. Okay, remember test tube number 7 was at 0 degrees Celsius, which is the really, really cold one. So if the color is lighter, it's because there's more greens or more blues originally mixed in with it. So you're going to be telling me which one worked the best out of these 3, which one worked the worst out of these 3. Which condition does amylase prefer to work best? Let me see. At the bottom here, you have a bunch of questions that you're going to be answering. So you're going to be telling me, what effect did cold temperature have on the enzyme amylase? Based on your results, did amylase digest starch at 40 degrees Celsius? And you can use your color results to explain that. Number 3, you're going to say, what effect did high temperature have? And then 4, you're going to answer the question as well.